good thing.
Oh, why don't we get
The one with
Say that about
tomorrow we're
yourself.
doing 4, just every day.
You just got a combo, so we're doing like 32 in one day.
Let's go ahead and learn some more about replication in DNA. So yesterday we learned about like how DNA was discovered. Honestly, not terribly exciting because it was a lot of people's names and stuff. Today it gets a little more exciting because we're talking about DNA itself. I think we're going to do the lab on Friday because it's just kind of a fun way to end the week on a Friday with a lab. So I think that's how we're going to end the week with a lab. And the lab is pretty easy, it's not too complicated, and it's a pretty straightforward lab. So, DNA in a single human cell, each one of your cells, so it's kind of crazy to think about. Every cell in your body has DNA. It is about 2 meters or 6.5 feet long. So it's really crazy to think about that every single one of your cells has a DNA strand that is 6.5 feet long. So how can that much DNA pack into a nucleus that is 10 micrometers in diameter? And the reason that it is able to do that is that the proteins that are associated with DNA, they help to keep it organized. So an actual DNA piece is taller than I am. I am about 5'9". So one piece of DNA is 6.5 feet, and think of how many cells your body has. And there is that much DNA in every single cell. So we have a lot of DNA, and the DNA really is that long. And when we extract the DNA out of the strawberry, you'll see the actual DNA. Because we separate it out of the nucleus, we dissolve the nucleus, and we'll see the DNA physically without a microscope. So it is pretty interesting. But the DNA gets all wrapped up nice and neat in a tiny little bundle, and that's how it is able to stay within the cell. Nice, tight, neat little bundle.
I'm 9.
Really?
I was taller than everybody except for like, the 6 footers. And Bryce at 5'6".
They bring in 6
That's probably a part you are, right, Bryce?
Yeah, 5 foot. They bring in 6
He is. What do you think he is?
Yeah, he's probably 6 foot.
Yeah, they usually just add a couple inches. Do they not?
That's only the really short people.
No, yeah, come on.
It means 6 foot.
And how does this do?
6'2", yeah.
Aren't you like 6 foot?
Nobody said you listed at 6'1".
No, I mean measure.
Exactly.
I'm not sure. I'm not sure.
So half an inch.
Look, my shoes not on 5'8".
Okay, so a
I
chromosome
think half an inch is a lot.
is the structure that consists of DNA, and it is wrapped up in a nice, neat little bundle. So typically you can't see the chromosomes unless a cell is getting ready to divide. Because really the best way to describe it is, imagine you're going to the store to buy some yarn, because you want to do a yarn project. I mean, I know you guys probably don't do yarn projects, but, just for the sake of the story. So you want to buy some yarn. If you want to transport yarn from one place to another, it helps if it's in a nice neat bundle, because if the yarn is unraveled, then you go to the store, you get the yarn, it's all unraveled, and you're trying to put it in a cart, and you're trying to take it up to the store to buy it, and then take it home. It's gonna leave a trail behind, and it's gonna get all tangled, and it's gonna be hard to get into the shopping cart, into a bag, and then once you get home, you can't really do much with it because it's all tangled up. But in order to transport it from the store to the cart to home, if it's wrapped up nice and neat, it's much easier to do stuff with. So when you get it home though, in order to actually use it, you need to unravel it. So that's how DNA works. In order to use the DNA, it needs to be unraveled. We can't use DNA if it's all wrapped up nice and neat. We can't use the yarn if it's all wrapped up nice and neat. We need to unravel it in order to use it. We only need to wrap up the DNA if we're transporting it from place to place. So the same is true for the DNA. If we are transporting the DNA, then we do need it wrapped up. So that's why we can see the DNA in the chromosomes during mitosis and meiosis, because that's when the DNA is being moved from one place to another. So we wrap up the DNA into chromosomes when we are transporting it from one place to another. Now, a histone is a little tiny protein that helps to wrap up the chromosomes and the DNA. A nucleosome is a linked DNA that wraps around a histone twice. So, the blue here is the protein. This is the histone. The nucleosome is this whole thing. So, we've got a histone, and then the DNA wrapped twice around it. So, this whole thing is a nucleosome, and the blue is the histone. So, when we have the histone plus the DNA twice, that's a nucleosome. When we just have the blue thing, that's a histone. Alright, so this is kind of what the chromosome looks like, and then we break it down and we break it down and we break it down even more. And so each one of the slides is going to break it down into smaller and smaller pieces. So chromosome, and it gets broken down until eventually we get to our nucleosome, which is down here. So our chromosome with our centromere. So this is the DNA wrapped up all nice and neat in a tiny bundle. So this is a chromosome, and this has been copied into what's called a sister chromatid. And we'll learn more about this when we get to mitosis and meiosis. So this is one chromosome, and this has made an exact copy of itself. So this is one, this is the exact copy that it's made. So this is it's broken down into its coiled, it's all nice and neatly organized. So if we uncoil it, it can be seen, this is it uncoiling even more, and kind of like beads on a string. So this here is a nucleosome. So the green is a histone, and it's a histone with 2 wraps of DNA around it. So the DNA is wrapped twice around a histone, and that creates a nucleosome. So we've got a histone, which is the green, 2 wraps of DNA, so the whole thing is a nucleosome. So nucleosome consists of part of a DNA molecule looped twice around a histone. So histone, 2 wraps of DNA, the whole thing's a nucleosome. And this shows the DNA. DNA in the double helix wrapped twice around a histone. So all packaged nice and neat together.
Grace, I heard you say yelling me to dunk in love.
Why didn't
See, nobody looked at you.
Why do you need a oop?
I need a loop.
Actually, it's easier to dunk
I did sure.
I don't know, it's good. All sex together.
At metaphase, the chromosomes are duplicated and are at their most condensed. In each chromosome, 2 identical sister chromatids are held together at a constricted region called the centromere. When a chromosome is condensed, interactions among chromosomal proteins keep loops of DNA tightly packed into a supercoiled array. On closer examination, we can see that the chromosomal proteins and DNA are organized as a cylindrical fiber. If we immerse a chromosome in salt water, this fiber loosens up to reveal a beads on a string organization. The string is one molecule of DNA. Each bead is a nucleosome that consists of a DNA molecule looped around a core of histones. Nucleosomes can loosen in controlled ways to give enzymes access to specific DNA regions.
They're really only
So as I was mentioning, we start off with what is called a chromosome. So the chromosome is one individual, and that is the genetic information. But when this material is getting ready to duplicate when we go through mitosis, which is the cell making a copy of itself, it needs to make a copy of its genetic material too. And in that case, it goes through replication, which is a process we'll learn about in a little bit. And it needs to make a copy of its information, and it makes a sister chromatid. In that case, we have the X shape, which is on the far right. And that is joined in the center by what's called a centromere. The centromere is in this instance here, we have one chromosome, and this is the sister chromatid. So one, and this is the copy. This is exactly the same as this one. It's made a copy of itself. So this one is exactly the same as this one, and together they make a sister chromatid. These are going to separate out during the division process of mitosis, and they're going to get pulled into individual cells. And so this is going to go to one cell, and this is going to go to the other cell, and that way the two cells are going to have exactly the same information in them, and so the cells will be identical to each other. The kinetochore is where the spindle fibers are going to attach during the mitosis process. The centromere is where the two sister chromatids attach. The kinetochore is where the spindle fibers are going to attach.
You don't have to. Let me come here.
It shows everywhere, no shot.
I have so many does, but I don't see my hunt.
I can walk around for 5 minutes and shoot 20 of them.
I can walk on the road and shoot a doe, but I can't run around and shoot a doe.
It's so many.
There's so many spikes.
In Fisher's Bottom,
You
there's
guys don't have any deer over there?
like 50 of them come out.
You don't
That
see
field
that
has
field?
so many deer. Every time I drive by, I'm gonna fish.
All right, so when the cell prepares to divide, it does need to duplicate its chromosomes first. And this is a process called replication. So the DNA needs to replicate its information. It needs to make an exact copy. And it's important that the information is exact. It needs to make an exact copy of itself. Because if that is a skin cell, it wants to make sure that the new cell that is formed is also a skin cell. It doesn't want to suddenly be like, oh, I want to be a tooth cell. No, we want to make sure that if it started off as a skin cell, it's going to end up as a skin cell. That is its job. It doesn't need to start as a skin cell and end up as something else. So it's important that replication ends up exactly the same as what it was. Now, there's another process called meiosis. In meiosis, we want something different. We will learn about mitosis and meiosis when we get back from Christmas break. That'll be our next unit that we learn about. But for now, replication, transcription, and translation is what this unit's going to be about. But replication has to take place first before mitosis can take place.
Make fun of me in science.
Science computer. That's what Dylan would do. Dylan would make fun of your coaching parents.
You're low key, no capping.
I like my dad.
That's correct. Hip grammar.
My dad told me I was capping the other day, and I was like, what? I was like, what are you talking about? I didn't even know you know what that word meant.
What are you doing? You're doing something wild. Today Bryce was doing something wild. He had a 6 in
I was sitting there.
in math class.
What are you 6 in?
You know what I'm talking about. Math class, he put a 6 in.
I thought 8 times.
Oh, yeah, yeah,
8
yeah.
times 3 equals 17. I was
Oh,
gonna-
yeah.
That's what it was.
Close, Chris.
But you thought 8 times 3 was 6?
No, it was
He
32.
had a 6 in there. That's all I know. It was messed up.
DG did try to say x times x is 9x.
No, I didn't know you did 9x. I didn't know you split it up into 3x.
So this is just showing the same thing. One chromosome and then two sister chromatids exactly the same, joined by the centromere. And that is going to separate out during mitosis.
Quadratic functions.
So the human body has 23 chromosomes, two sets, that 23 from your mom, 23 from your dad, so for a total of 46. So two sets of 23. 23 from one parent, 23 from the other parent. We are termed diploid because we have two sets, die for two. Now, the sperm has 23, the egg has 23. So those are called gametes. The sperm and the egg are gametes, or the reproductive cells. They're also called haploid because the sperm and the gametes have half of the information. Now the rest of the body cells, those are somatic cells. Body cells are somatic cells. They are the cells of the body that have a full set of information. So pretty much all of the cells in your body are diploid, having a full set 46 chromosomes. So there's something called a karyotype. This is an image, like a snapshot of the cell showing the 46 chromosomes. So they used to take a picture under a microscope of the cell when it is going through mitosis. Now it can be done in a computer, and it still takes a picture of the cell going through mitosis, but now the computer can do it and do it much better and kind of zoom in, and it arranges it in such a way that it can match up the chromosomes. Now, typically you can't tell which came from which parent, unless you also have the parent's karyotype to compare it to. So if you know this is the mom and this is the dad, you can get the parent's chromosomes and then you can compare. And then you can usually figure out which came from which parent. But if you're just taking like the individual, like if you just took my chromosomes, you wouldn't be able to tell which of my chromosomes came from my mom and which of mine came from my dad. But you would be able to know, okay, one of these is from one parent, one of these is from the other. You just wouldn't know which is which without testing my parents. But a karyotype is an image that matches up the chromosomes and kind of helps you get a visual to compare them. But humans have 46. Different animals have different numbers. Different plants have different numbers. You could have more, they could have less. It varies depending on the species. If it is alive, they have chromosomes. Things that are non-living, like our desk or our chairs, do not have chromosomes. Unless it's made of wood, then it has chromosomes too, if it's real wood. If it's plastic, no chromosomes. If it's wood, chromosomes.
It will snap my computer in half.
I might know because.
Blood is collected from the person being analyzed. The blood is added to a growth medium that also contains a chemical that stimulates mitosis. The cells are allowed to grow in this medium for 2 or 3 days at body temperature. Colchicine is added to arrest cell division at metaphase. The arrested cells are transferred to a centrifuge tube.
It's important to note that metaphase is the phase that is used because that's when the cells are the most condensed and most packed together. So metaphase is, they stop the process at metaphase because that's when everything is really tightly compacted.
Centrifugation concentrates the cells at the bottom of the tube. Addition of a hypotonic salt solution causes them to swell up and move apart. The lysed cells are prepared, fixed, and placed on a microscope slide. A cell is observed and photographed under the microscope. The part of the photo that shows the cell's chromosomes is enlarged, and images of individual chromosomes are cut out. Finally, the images are arranged so that all pairs of homologous chromosomes are horizontally aligned by centromeres. The result is a karyotype diagram of the arrested metaphase chromosomes.
So this is important because this will help determine genetic diseases and disorders as well. So each of these, this is from one parent, this is from another parent. You can see they're similar sizes and similar shapes, and the centromeres is in a similar location. So usually the last one is the male and the female chromosome. So in this case, they're different sizes. The X chromosome is typically larger than the Y chromosome, so in this case, this would be a male because they have a Y chromosome and an X chromosome, so this would be a male. For Down syndrome, it's usually called trisomy 23 because typically at the 23 chromosome, there'd be an extra. So they have an extra chromosome, and that's what causes Down syndrome. They have 3 chromosomes instead of 2 chromosomes, and that's what causes Down syndrome. So there would be an extra chromosome around here instead. So that's what causes Down syndrome. They just have an extra chromosome. Instead of 2, they have a 3rd one.
Miss Eric, if you're a scientist, is it possible for a 5'7" human being to be above the height of 6 foot, even though they're 15?
Yes.
There is a lot of height variation, and
Anything's
So,
possible.
yes.
With your mind to.
Like, if your parents are tall?
yes. There's a family, I think on TikTok I saw a family, that the siblings are like really, really tall.
Oh,
One
I've
kid's
seen that
like
family.
7
One of them's like,
Yeah.
yeah, they're all giants.
They're all like 7 foot.
They're so weird too.
Yeah, they're all
Yeah.
like, they all weigh like 120 pounds.
Yeah.
They're all,
Super
like,
tall,
really
super
skinny and
tall,
really tall.
Super weird. He's like-
So here's another example of how, here's another example of karyotype and how it matches up. Again, the dark bands are representing different genes. So you can see how the genetic code matches up. So not only are the chromosomes matching up, but different genes match up. This will get important when we talk about genetics because we will have a genetics unit as well. And so these are different genes. So this is a chromosome from the mom and this is a chromosome from the dad. And you get different genes inherited from your parents also. Again, this individual would be a male because they have an X and a Y chromosome. And anytime a Y chromosome is present, that means they're a male. If this was a female, they would have 2 X chromosomes. So some chromosomes are larger and some of them are smaller, but one from mom and one from dad.
See, my mom's taller.
All right, so autosome is a chromosome that is the same in males and females. So a lot of these are like coding for the body. So hair color, eye color, height, things like that. So they have the same length, same shape, and the same centromere location. And they hold the information about the same traits. So again, usually about the body. Hitchhiker's thumb, whether you have a straight thumb or a hitchhiker's thumb, all of those kind of traits, those are autosomes. So characteristics that are found on the 22 other chromosomes. This last chromosome again, that's the XY chromosome, that determines male versus female characteristics. So autosome are the 22 other chromosomes. The XY chromosome is usually not the autosome. But they're about the same length, the same shape. You get one from mom, one from dad, but they have about the same information. So you get like a chromosome from hair color from your mom, a chromosome for hair color from your dad. And the way those two interact will determine what your hair color actually is.
Do you have hitchhiker's thumb?
What?
Do you have hitchhiker's thumb? Is that where it
I do
bends-
not. Mine is very straight. It doesn't get much straighter than that.
Where it bends back super far like mine.
Yeah, you definitely have. Oh my goodness. Yours is hitchhikers. Mine is not. Mine is
that-
about as straight. Yes, that is a hitchhiker's. Mine is about as straight as they come. Yeah, no, mine is straight. I cannot. That's as far as it goes. I kind of wish I had a hitchhiker's. So I was straight too.
I always thought this was the
No,
like,
mine is. I can't do it. Do it.
you
I want it. I think it's cool.
I had a friend who would always just
Oh my
like,
God,
just
you're
put me in class.
You should have watched Monster.
It doesn't do it. It's boring. I want a hitchhiker's
Hey, can you do
thumb.
this?
I was like, do it today.
Doesn't work.
Look, okay.
Also, it's not
Turn the other way around.
Mom, you didn't even do anything.
Maybe if there's actually some.
I had somebody say my thumb was abnormally long.
Tyler convinced 215.
And I think it's just because it's straight. And it doesn't bend back.
You finished, Tyler?
I have not benched in months.
last time I did was
I
155.
don't think it's that long.
Oh my god. How does the body work? I'm so.
He eats such a healthy lunch every day, and that really contributes to it.
All right, so the sex chromosomes, in order to be considered female and have female characteristics, it's the XX chromosome. And the only way to have male characteristics is the XY chromosome. So, in determining offspring, the male is the one that determines what the offspring is. So, when producing a child, it is always the male that determines the offspring. So let's say that I wanted a girl. I have no say in the matter because I can only give an X chromosome because I have two X chromosomes. It is my husband that determines the offspring of my children, whether they are male or female, because he can give an X or he can give a Y. I can only give an X because that is all I have to give. I have two X's. That's all I have to give my children. They get an X or they get an X from me. My husband can give an X or a Y. So he gets to determine whether I have a girl or a boy. That's just kind of how genetics works. Thankfully, I got one girl. My husband's family, they all, his whole family, it's just, they all have boys. So I was very grateful that I got a girl because it's all boys. They're all boys. So I'm glad I got a girl.
At least that means your name can be passed on.
That's true.
I'm the last in Grindstaff.
Really?
Yeah.
Well, see, then you
So it might be rough.
Easy, all the pressure's on you.
Yeah.
all pressure.
Yeah, leg.
Now I've got 3 boys and 1 girl.
I was the last at Thornhill line, but guess what?
Keaton? No, he's amazing.
No. And Uncle Jared, he's having another kid.
Is it a boy?
Yeah, he had a boy, Silas.
Silas is cute.
I know what I'm naming my kid.
And
If I have a boy.
hey, he's 40.
Named to be Coit. Sweet name.
Wait, I like
Yeah.
Wait. I'm gonna take that. I'm gonna have to pick first and name my kid.
You can't take my name.
Yeah, I can. Yes, I can.
Hey, Jerry, you gonna live in Harrisburg when you're older?
You should have like a.
I think you want to go to Maine.
Probably.
You should name your kid
I'm going to Michigan.
I'll try to
Or
leave, but I
Mississippi.
won't be able to with my parents.
Why?
No, they said they're as soon as me and my sister move out, they're getting a house in Florida. They're leaving.
Now, not all animals rely on the XY trait. Some animals will use temperature. For instance, reptiles will use whether they lay their eggs close to the surface or deep down. If it's colder, if it's warmer, that will determine if it's a male or a female. So if they want more girls in their population, they'll make their temperature one way. If they want more boys in their population, they'll make their temperature another way. So they can actually determine if it's male or female based on temperature. Which is great until you get to global warming. And so global warming is swaying the population, making more of, I think warmer is male. So they're having more males in the population because the temperature is warmer, and so that is swaying the population one direction. At least with XY chromosomes, you get a 50-50 chance that way. So.
Didn't they make a robot that can determine what child you have, who they are?
The way technology is coming now, there is artificial insemination and IVF treatments and in vitro fertilization. There's becoming more advanced that you can pick and choose more now. But
Oh yeah.
as far as picking and choosing whether you have a boy or a girl, there's more argument on that. But whether if you pick or choose if your child has cancer,
Dude, it runs in the family.
see?
You know what's
So-
in my family? Heart disease.
Would you rather pick or choose if your child is a boy, or would you rather pick or choose if your child has a genetic disease? Now that, if I could choose whether my child had cancer or not, no, I don't want my child to have cancer. No, I would choose no.
No, right? Said I died.
65. All right. So that's all the notes we're taking today. You guys have the rest of the time to work on the assignment. I think most of you are pretty close to done on that. So notes took a long time yesterday, so I'm giving you a little bit more time to work on the assignment today. You will get a new assignment tomorrow, though. So this is the last time to work on the whodunit assignment today.
Wait, who done it, Mom?
So you'll get a new assignment tomorrow, And then we'll do a lab on Friday. So we will get a lab on Friday. Any questions? Okay. All right. So lab on Friday, new assignment tomorrow.